This will be a two-part lesson series. In the first part, we'll review what we know about DNA and some of the early history of life on Earth and how early organisms used DNA to build proteins and then duplicated DNA for purposes of reproduction. And then in the second part, we'll introduce the concept of a cell cycle, that all cells on Earth have what amounts to a moment of birth, a period of growth, and then reproduction, where the parent cell divides into two daughter cells. First, let's remind ourselves that cells are composed of molecules. So here we see a simple bacteria type of cell conceptually laid out here with different categories of molecules that make it up. For example, the membrane is composed of lipid molecules. It makes the barrier of the cell. And the lipid molecules, the tails of the lipid molecule, are a chain of carbons and hydrogens, and that part of the molecule is a fat molecule. We know that all cells must get energy from their environment, and lots of cells import sugars, like glucose. The chemical bonds of glucose store lots of energy, and inside the cell, proteins will break those chemical bonds and repackage that energy in molecules of ATP. And then we have two important molecules down here. We have proteins and DNA. Proteins, of course, are the workers of the cell. They're the ones that perform the chemistry of life. Well, proteins are chain molecules composed of amino acids, and there are 20 different kinds of amino acids found in proteins in living things on Earth. Now, there are 20 different building blocks, you might say, but you can put them in a huge variety of different sequences. The sequence of those 20 amino acids determines how the protein folds up, how this chain folds up, and the three-dimensional shape of the protein determines the job it can do, what kind of chemistry it can do. A protein may be as little as a few amino acids stuck together, or some proteins are hundreds or indeed thousands of amino acids in length. Over here we have DNA, the double helix. When we untwist it, we'll see the two strands here, and we have learned that the sequence of these DNA bases, these DNA letters, instructs the cell to build a protein with a certain sequence of amino acids. So this is how DNA can store information. The sequence of these DNA letters determines the sequence of amino acids in a protein. Now let's introduce a couple of terms here. We're going to identify what a gene is here, is the DNA recipe for making a protein. That's called a gene. And we can see a gene right here. This stretch of DNA in our diagram is supposed to represent the information to make this particular protein, this blue protein. Now, we've learned that there is an intermediate molecule. Messenger RNA is a temporary copy of the recipe of DNA, and it's the one that hooks up to the ribosome, and the ribosome will actually put together the amino acids to make this protein. But we want to distinguish between the DNA molecule itself and what a gene means. A gene refers to the information content that is stored in the physical molecule of DNA. So DNA is a physical thing, it's a molecule. A gene refers to information, the information to build this protein. Now, in bacteria, the DNA is in the form of a circular chromosome. So we're going to identify a chromosome then would be a longer bit of DNA that has multiple genes. So in fact, every protein in this cell here has its corresponding gene. So the gene for the blue protein might be right here. The gene for this green protein might be right in this bit of DNA. The gene for this protein might be over in this bit of DNA. So every protein has a corresponding gene in the DNA somewhere. And in fact, all of the genes combined is called the genome of the organism. Now, we've learned in the early phase of life on Earth, bacteria colonized the ocean, they reproduced, and they evolved. And one kind of evolution that we looked at was the evolution of a new capacity to utilize a new food source. So bacteria had to get their energy by consuming molecules in their immediate environment. Well, we told a story where early on the red bacteria here could only eat the green food, but in time they evolved, the population evolved the capacity to eat not only the green but also the orange food molecules. And the way to understand this was this concept of gene duplication, that sometimes during DNA duplication, big mistakes happen where an entire gene gets duplicated. So here we see the gene that makes the transporter protein that allows the green food in. If it gets duplicated, then the cell has 2 copies of that same gene. Now, early on, that might not really make a big difference to survival, but in time, this duplicate gene then would be free to accumulate mutations. You see, if you only have 1 copy of the gene, then you go mutating this one, then you might mess up this protein. But if you have a duplicate copy, now mutations can accumulate here, which can alter the protein made from that mutated gene. And we saw that, again, just by random mutations in this duplicate gene, some of those mutations might make a protein that allows for the importation of a different food molecule. And so then you get the situation where now the bacteria can eat not only the green food, but also the orange food. So this then is an explanation for how the bacteria population could evolve the capacity to eat new food sources. Gene duplication opens up many evolutionary opportunities. We should also note, though, as gene duplication events happened, the genome was increasing. So the organism now has more and more genes. So it's getting more and more DNA over evolutionary time. We also learned that bacteria are restricted in how big they can grow. They face an energy problem as they grow larger. Recall, the bacteria has proteins in its cell membrane, so the energy production is proportional to the surface area of the cell, but the energy needs, or the energy used, is proportional to the volume. All that chemistry going on in here requires energy. So as cells get larger, the surface area grows more slowly than the volume, so cells will face an energy limitation. We learned that a very important event happened some 2 billion years ago where 2 cells merged, and this, we said, was called endosymbiosis, and it solved the energy problem. It created a situation where you could have smaller cells that were good at producing energy, reproducing inside the cell and providing the larger cell with lots of energy. And so that allowed those cells to grow even larger and more complex. And we think this endosymbiosis event happened because of the evidence that these mitochondria, as we now call them today, they're the size of bacteria, have DNA like bacteria, and they reproduce within the cell like bacteria. So endosymbiosis solved an energy problem, and it allowed for cells to evolve to be more complex and larger single-celled predators, more active predators like amoeba, for example. Now, of course, you can imagine as you become a larger cell, you may need new kinds of ways to operate a larger cell. And so new genes were evolving in these new larger species. For example, the genes that code the proteins for pseudopod control and just moving in general and responding to the environment like these active hunters do. So new genes were evolving in these types of single-celled organisms. And with new genes evolving, then the amount of DNA is going to be increasing again for these organisms. And so organisms are going to increase their genome size over evolutionary time. So we go from bacteria with a genome size on the order of thousands of genes to larger cells with even more genes tucked away in their nucleus. Now, we have learned about the physical structure of the DNA molecule, but as cells became more complex and accumulated more and more genes as a result of gene duplications, the organisms had to find some way to handle all of this DNA. And one way that the cells did this was to package up their DNA, to wind up their DNA around proteins. We'll call this DNA coiling. And this coiling then allowed cells to pack lots of DNA into a small volume. So as organisms evolved to be larger and more complex, they needed more genes to run these larger, more complex cells. And so in the nucleus then, a way had to be invented to package more and more DNA into the small volume of the nucleus. And one way to do that was to coil up the DNA. This is actually a picture taken from a light microscope, 1000 magnification. This is a picture of DNA chromosomes from a fruit fly. So what we're witnessing here is the coiled nature of DNA. In fact, it's been stained in a way that we can see these bands of red and white bands. These would be individual genes, but they're in the coiled up phase. So it would be, what we're looking at is this kind of structure right here, the supercoiling of DNA. Now, in these large complex cells, again, the DNA is stored in the nucleus. Here we have some paramecia here. Again, using certain lighting techniques. The blue structure here is the nucleus of the cell. Here we see it enlarged here, and inside the nucleus then would be all of the DNA necessary to build all the proteins for the paramecium. In part 2 then, we'll take a look at how these complex cells handle their DNA, both during the growth phase and then what they do to make sure that the daughter cells have a full complement of DNA.